A very good morning to everyone. So I am Bushra, your botany faculty, and today I am going to take this next session. I hope your brains get warm up. Make sure that you stay rooted because in this session we are going to branch out. Now let's start with this session. So here in this session I am going to discuss the question from AIATS Plus Phase 2 Mock Test 5 which was held on 16th of February 2025. So in this particular session I am going to discuss the question particularly from these topics in which most of the students had made mistakes. So first two topics, that is molecular basis of inheritance and biodiversity and conservation, are from 12th class, which are very important. This chapter holds very great value in the NEET exam because from this chapter only 6 questions had been asked. Okay, so this chapter is really important. Now these 3 chapters, biological classification, anatomy, plant growth and development, are from the 11th class and these are also very important. Now let's start with the very first question of this session, which says dicot stem can be differentiated from monocot stem as the former contains. So this question is basically asking about the stem, the differentiating between the dicot and the monocot stem, and this question is from the anatomy of flowering plants. Now first of all let's see how they look like. So अगर आपके सामने ऐसे दो डायग्राम आ गए then how will you distinguish between कि इनमें से कौन सा dicot है और कौन सा monocot है. So if we see the transverse section of the dicotyledonal stem, then it will look like this. That means it will have a very huge area in between them which is known as, let me select the pen, yeah, so this is known as pith. So अगर ऐसा structure दिखेगा then it is dicot stem. If the transverse section of any particular plant section look like this, then it will be monocot stem TS. Okay, now let's start differentiating in between them. So अगर हम epidermis की बात करें, which is the outermost layer, सब में single layer होती है, yes, it is common in between them. Now let's talk about the next layer, that is hypodermis. Hypodermis, which is present next to the epidermis. If we talk about the dicot stem, तो उसमें collenchymatous होती है ये. It is collenchymatous, that means collenchyma are present in the hypodermis. If we talk about the monocot stem, in them hypodermis is sclerenchymatous. Right? If we talk about monocot stem, here the ground tissues are not well differentiated. That means unme ground tissue are not differentiated in terms of endodermis, pericycle, etc. Undifferentiated ground tissue is present. But if we talk about dicot stem, it has very well differentiated ground tissues. So if we talk about endodermis, which is actually the innermost part of cortex, endodermis, it is basically barrel-shaped cells. Barrel means जैसे barrel होता है ना, the curved from the side and having this type of structure like this. It is basically composed of barrel-shaped structures, cell structure, and it have one special quality, or we can say characteristic feature, that means they have deposition of a starch grain. जिसके वजह से it is also known as starch sheath due to presence or deposition of starch grain in the endodermis layer. Okay, please remember that this is the differentiating feature for the dicot stem. If we talk about the pericycle, it is single layered in the dicot stem. Now let's come to the vascular bundles. I am writing VB for that, vascular bundle. If we talk about dicot stem, तो you can see that vascular bundles are arranged in a ring. ये जो structure दिख रहे हैं, these all are vascular bundles and they are arranged in a ring arrangement. This is also the characteristic feature of dicot stem. And if we see the monocot stem, we have scattered vascular bundle. पूरे stem के area में vascular bundle are scattered. They are not arranged in any particular form. We talk about vascular bundle again, they are conjoined, right? And they have the presence of cambium. That means they have the capability to undergo secondary growth. Cambium is present. If we talk about monocot, so here no cambium is present. That means they have conjoined and closed vascular bundle because they do not have cambium. Right, so this is the case. If we talk about medullary rays, so the cells, the parenchymatous cells which are present in between the two vascular bundle will form medullary rays. So this area is actually composed of cells which will form medullary rays. But if we talk about monocot stem, there is no such medullary rays are present. Here, if you see the vascular bundle arrangement, the vascular bundles which are larger in size are present at the center, and the vascular bundles which are smaller in size are present towards the periphery. So this is how it actually look like. Right, so and pith, as I've told you initially, that it has very large pith. Pith is present and it is large. Here pith is absent, you can see that there is no pith. So these are the differentiating features between them. One more feature which is the characteristic feature for the monocot stem is presence of water cavities within the vascular bundles. I am writing here water cavities in vascular bundle. VB here stand for vascular bundle, please remember that. Right, so this is the characteristic feature for the monocot stem. ऐसे questions भी आते हैं that water cavities are found in monocot stem, dicot root, monocot root. So the correct answer is monocot stem. Right, so this is how we learn the structural differences between the structures. Right, now let's get back to the question. Let's see the options. The first one is water containing cavities. जैसा कि क्वेश्चन में पूछा है, हमको दोनों स्ट्रक्चर को डिफरेंशिएट करना है एंड वी हैव टू चूज द ऑप्शन फॉर फॉर्मर, दैट मीन्स द ऑप्शन फॉर डाइकॉट स्टेम. तो फर्स्ट ऑप्शन है वाटर कंटेनिंग कैविटीज विच आर एब्सेंट इन डाइकॉट स्टेम बट दे आर प्रेजेंट इन मोनोकॉट स्टेम. सो ये ऑप्शन इनकरेक्ट हुआ फॉर द डाइकॉट स्टेम. राइट, नाउ लेट्स प्रोसीड फर्दर विद द नेक्स्ट वन विच सेज स्टार्च शीथ. So yes, starch sheath कहाँ present थी? Please comment में बताइए कि किधर है starch sheath present. As I am not able to see any comment on the screen, there must be some problem. Please check over the screen if there is. Now let's see that option 2 which says starch sheath. So starch sheath is present in the endodermis, which is the innermost layer of the cortex. Right, due to presence of a starch grains. So yes, this is the differentiating feature between the dicot stem and monocot stem. Let's see that options also. Option 3, sclerenchymatous hypodermis. Sclerenchymatous hypodermis is present in the monocot stem, not in dicot stem. Dicot stem में collenchymatous hypodermis थी. Right, so this option is also incorrect, and the fourth one is scattered vascular bundles, which is found in monocot stem, not in dicot stem. So this option also get ruled out. So this is how we rule out the option and hit the correct answer. Right, now let's proceed to the next question, that is question number 122, and this question is from the chapter molecular basis of inheritance. Okay, Vikram, yes, very good. Yeah, dicot stem, dicot endodermis, yes. Now let's proceed to the next. Anushka is asking, what is starch sheath, ma'am? Can you please explain? So starch sheath is basically another name for the endodermis layer. Endodermis क्या होता है? It is the innermost layer of the cortex where starch grains are present, where deposition of starch grains are present, due to which it is known as starch sheath. I hope I made you clear that what starch sheath is, and it is specifically present in dicot stem. Monocot stem में नहीं होता. I hope I made you understand. Okay. Now let's get back to this question, which says DNA replication is said to be semi-conservative because. question is about semi-conservative nature of DNA replication. So what you understand by DNA replication? It is basically making the duplicate copy of DNA. Okay, Anushka, you understood. Very good. Now, let's see the process of DNA replication. So, the model of DNA double helix was given by Watson and Crick. Once they proposed this model, a double helical structure of DNA, after that they have proposed that they replicate semi-conservatively. So, let me write the scientist name for you people. That Watson and Crick are the same person who gave DNA double helix, and उन्होंने ही बताया था that they divide or they replicate in a semi-conservative manner. But it is proven by another scientist known as Meselson and Stalin. They basically experimentally proved that DNA replicates semi-conservatively. How? Let's see. First of all, what is their genetic, or we can say the material which they have used in their experiment is prokaryotic organism, that is E. coli. Right? So this is their experimental organism. Yes, Bemker Karmakar experiment was done by Meselson and Stalin. Very good. Now, what they did, they basically make their culture, they basically put this bacteria into the culture media, which consists of NH4Cl, ammonium chloride. The medium consists of ammonium chloride, which is the only source of nitrogen in the medium, which are required by the E. coli for their growth and development, or we can say the replication. But the twist is that they put heavy isotope of nitrogen in them, that means N15. Chemistry आपने ज़्यादा अच्छे से पढ़ी है, you must be knowing what are heavy isotopes of nitrogen. So he basically used heavy isotope of nitrogen, which is N15, right? And after some time he observed and extracted the E. coli. अब extract करने के लिए उसने extract कर लिया, but how he will observe that it got incorporated in them or not. Then he made a centrifugation tube. How he made? He take one tube in which he put cesium chloride, CsCl, right? And in them he centrifugate this particular compound in them and made the gradient in which he made that heavy cesium will be at the base and lighter one will be on the top. Like this, the gradient will be formed, right? As it is centrifugation, it is basically separated on the basis of their weight, right? So this is how the gradient is made. Then this Meselson and Stalin, what they did, they took the culture medium. Yes, CSCL uses density gradient. Absolutely correct. So what they did, they took the E. coli from this medium and put them in this. And then they observed that the band is coming here at the lower side. That means E. coli is having substance which is heavier. As they have used the heavy isotopes of nitrogen, that is N15, तो लाजमी है कि वो सबसे नीचे base पे accumulate होंगे. So here they observed that the DNA or the genetic content in the E. coli is having N15 in them. If we talk about the two strands, so both strand is having N15, right? Later on, what they did after 40 minutes, this one is done after 20 minutes. I'm writing here, this is done after 20 minutes. Later on, after 40 minutes, they put the E. coli in the media containing N14, the light nitrogen. सबसे पहले उन्होंने क्या use किया था? Heavy isotopes of nitrogen, that is N15. Next they use N14 and then allowed them to divide. N14 में किनको डाला, which are having this one, the heavy isotopes of nitrogen, N15. Later on, after once they have extracted after 40 minutes, then they observed that hybrid strand, that means N14 and N15, N14 and N15. This is observed. That means the two strands of DNA, we talk about E. coli, it got separated, right? One is having the parental strand and another is newly synthesized. The 15 one is parental one and the 14 is newly synthesized, which got replicated during this medium. So this is how they experimentally proved that how DNA replicate semi-conservatively. That means during the DNA replication, the DNA template, one will become from the parent and another one will be newly synthesized. For example, I will represent it from here like this. If this is red, strands of DNA will get replicate, then replication is taking place, one strand each from the parent and other strand which will be synthesized new are made from the another color. So this red one are from parent and another one are newly synthesized. That means in the daughter cells, 50% of the characters will be inherited from the parent as such. So this is how we say that DNA replicate semi-conservatively. Okay, so this is how they experimentally prove this thing. Now let's get back to the question that why DNA replication. Arbina is asking, ma'am, does hybrid remains constant throughout many generations? अगर हम same medium में रखेंगे तो yes, ऐसा ही होगा. Now let's see the options. The first one is only one of the two strands of a DNA is replicated. This does not explain that why DNA replication is said to be semi-conservative, so this is incorrect. Option 2, only half of the length of DNA is replicated. This is not the case, that why DNA replication is said to be semi-conservative. Option 3, each daughter DNA has one parental and one newly synthesized strand. So yes, it is absolutely true. जब भी DNA replicate होगा तो daughter में, every daughter DNA will have one parental and another one will be newly synthesized strand. This is how we know, we call them as semi-conservative. Option 4, both the strands of the daughter DNA are newly synthesized but are complementary to parent DNA strands. So no, this is also not the case. So this is how we understood the concept of semi-conservative. All the options are quite close or similar to each other, but if you know the experiment and know that how the DNA replication process have been proven, so this is how we can hit the correct answer. Now let's proceed with the next question, that is again from the molecular basis of inheritance. Question number 123, which says identify the correct statements and choose the option accordingly. As I can see, the statements are basically based on the DNA replication process, right? DNA replication, just हमने previous question में पढ़ा तो semi-conservative nature of DNA replication. Now we will see what and how does DNA replication proceeds in a cell. Right, now tell me what are the location of DNA in eukaryotic cell. DNA हमको nucleus के अलावा और कहाँ मिलता है? Write down in the comment section. Meanwhile, I am writing heading over here. Okay, so DNA replication. So if we talk about prokaryotic cell, so prokaryotic cell do not have any compartments in which the process could occur. So DNA replication in the prokaryotes will take place in the cytoplasm. If we talk about eukaryotes, as DNA is found in the nucleus where DNA replication will take place. Along with that, two other sites are also there Yes, Hema Malini Shetty, yes, you are very correct. Mitochondria. उसके अलावा भी एक और ऑर्गेनेल है जहाँ पे डीएनए प्रेजेंट होता है. Tell me that. Chloroplast and mitochondria. Yes, absolutely true. Very good. I like the students who are attentive in the class. Okay. Now, let's see. So, these are the locations where DNA replication will take place. So, if we talk about the cell cycle, so in the cell cycle, when does DNA replication will take place? Tell me. During cell cycle, during which phase DNA replication will take place? Please write down in the comment section. You must have studied the cell cycle chapter. Okay, in a meanwhile, let's proceed. Now, let's see what are the ingredients of the DNA replication process. As DNA replication is very complex process, it must be required certain ingredients, or we can say the requirements of the process. The first one is ori, origin of replication. What does it mean? Replication does not start randomly anywhere on the DNA strand. Yes, Bikram Karmakar, very true. S phase. During S phase, DNA replication takes place. Very good. Arbina and Hema Malini Shetty, very good. Okay, now let's see the requirements. So the first requirement is ori. Ori is origin of replication, from where the replication will initiate in the DNA strand. Right, so these are the specific locations which are recognized by the enzymes. If we talk about prokaryotes, so in prokaryotes, we have only single ori. I am writing P and small k for prokaryote, where single origin of replication is present. If we talk about eukaryotes, I am writing EK. Please don't get confused with that. Here, multiple origin of replication is present. Right, so first requirement is origin of replication. Now, here replication में हो क्या रहा है? It is basically making the complementary strand of the DNA. दो strands हैं DNA के, हमको इसको replicate करना है, मतलब duplicate करना है. इसका भी एक double बनेगा, इसका भी एक double मिलेगा. That means we have to make the complementary strand with the same, with a complement base pair, right? That means we require certain substrates which are known as DNTPs. What are DNTPs? These are deoxyribonucleoside triphosphate. Deoxyribonucleoside triphosphate. Now you will ask, then why we are not using deoxyribonucleotide, right? Where sugar is present, phosphate group is present, along with that base is present, that is complete structure is known as nucleotide. But here I have written deoxyribonucleoside triphosphate. That means along with one phosphate, two other phosphates are also added by the action of enzyme phosphorylase. So why basically this has done? Why we have added two more phosphate? Because this will act as a substrate. I think this color is not visible. Yeah, this will be. This will act as a substrate. Along with that, it will provide energy to this process, as this is very energetically expensive process in the cell. Substrate plus it will provide energy to the process. How? When the phosphate group will break down, then high amount of energy will be released in the form of ATP, which will be utilized in the process. So this is how it will act or help in the process. Now, let's proceed with the next ingredient. Oops. Where we have went? Okay. यहाँ पर आगे. So ये हमारी second requirement थी. Now let's proceed to the next requirement. Let me erase this area for some space. Okay. Now next requirement is certain enzymes. अब हमको enzymes चाहिए for this process. There are numerous enzymes which are required. Let's go one by one. The first enzyme which will be required during this process is helicase. As the name itself suggests that it is going to unwind the double helix structure. Next enzyme which will come into play is topoisomerase. Next one is not an enzyme but certain types of proteins which will help in stabilizing the single-stranded DNA. Those are known as SSBP. Those are single-stranded binding proteins. Please write down single-stranded binding proteins, right? Now, next enzyme is primase, which will help in initiating the process. Next enzyme which is very important, or we can say the master enzyme of this process, is DNA polymerase, which is basically going to proceed this process very fast. Now next enzyme is DNA ligase, which has the capability to glue the two strands. So these are number of enzymes which are going to act in this process. Now let's proceed into the process in which enzymes are required. So this is the image of the DNA replication, but we will come later on this. First start. As I've told you that in eukaryotes multiple origin of replication is present, so it will somehow look like this. Here bubble will be formed by the action of DNA helicase, like this, right? Multiple origin of replication hai. Here we are talking about eukaryotes, but if we are talking about prokaryotes, it will look like this, right? Bubble दोनों जगह पे बन रहा है, but in this, how we are going to understand? अब इतना complex process तो नहीं समझेंगे. To simplify the understanding, we are going to study this much, half, like this. ठीक है, so तभी यहाँ पे replication fork बनता. That's why it is known as replication fork. Don't assume that it is starting from the one point and ending at the another point. No, not like that. It will start like this. It will form simple bubble like this due to the action of helicase enzyme, and then replication will start in the both the direction. So this is the case. Now let's start. I hope you understood that how this structure has been come. Let me erase. I hope you understood that how replication fork is formed. Now let's see the action of first enzyme, that is helicase. Helicase, what does it doing? It is unwinding DNA double helix, and it require ATP. As I've told you that it is highly energy dependent process, so this enzyme require ATP to function. Once the helicase came and unwind the DNA double helix structure, as DNA की जो capability है वो double stranded में रहने की है, but अब हमने इनको single strand में कर दिया है. Then to stabilize them, we have certain proteins which are known as SSBP, single-stranded binding proteins. These are basically stabilizers which will stabilize the single-stranded DNA, right? So they will come and attach. Here, so that they do not get bind and form double-stranded structures like this. But once you have opened the DNA, then there must be certain supercoiling which will form here to the other side of the DNA strand, right? जैसे कि हम किसी rope को खींचते हैं. For example, if you take the braid and put your finger in the hair braid and do it like this, you will observe certain supercoiling on both the side, right? To avoid that supercoiling, another enzyme will come that is known as topoisomerase. If we talk about eukaryotes, it is known as topoisomerase. If we talk about prokaryotes, then it is known as gyrase. Right? So these type of questions are also asked, that supercoiling in the eukaryotic cell is done by which enzyme, topoisomerase or gyrase? Here specifically eukaryotes is written, then answer would be topoisomerase. So you have to be very careful in the question that which type of cell it is talking about, eukaryotes or prokaryotes, right? Now, after topoisomerase, as supercoiling has been controlled, the DNA helix has been opened. Now, next, what will happen? The action of DNA polymerase. But DNA polymerase cannot initiate the polymerization by itself on its own. खुद से वो polymerization नहीं start कर सकता. For it requires certain stepping stone to start the DNA replication process, and that stepping stone will be provided by enzyme primase. What does this enzyme will do? It will form the primer. What is primer? It is the strand of RNA. Remember that DNA में यहाँ पर RNA बन रहा है to act as a primer, right? And it is very small stretch of nucleotides, which is generally 10 to 15 base pair long, right? So here, to initiate the process of replication, stepping stone is kept by primase enzyme by putting primer in the initiating position, and then DNA polymerase will act. Next enzyme here, the master enzyme of this process, DNA polymerase. So if we talk about DNA polymerase, the complete name of it is DNA dependent DNA polymerase. That means by depending on the DNA, it is polymerizing DNA. That means template भी DNA और उसका synthesis भी DNA है. Okay? And the activity of DNA polymerase enzyme is from 5' to 3'. Remember this, it is very important that उसका direction किस side में होता है? It is from 5' to 3'. That means template का strand कौन सा होगा? 3' to 5'. Okay? Now let's see the diagram. In this diagram, where the DNA polymerase will come? Here first, primer will be formed, right? Next, DNA polymerase will come and start polymerizing the nucleotides complementary to the template strand, right? And then it will proceed in this direction, that is from 5' to 3'. The newly synthesized strands are forming from the 5' to, the polarity of them is 5' to 3', but the polarity of template strand during the DNA replication is 3' to 5'. Please don't get confused. The activity of DNA polymerase is from 5' to 3' over the template, which has the polarity of 3' to 5'. Okay? So this is the case that how DNA polymerase will come and act. So this is, we are talking just about 3' to 5' position. Now if we see the another strand where the polarity is just opposite, that is 3' is there, 5' is here. And the polarity of this enzyme is from 5' to 3'. So अब क्या होगा? The primer will come and attach here first. For example, forget this and assume that this much amount of DNA is just unwinded. So here primer will get attached, right, from the primase enzyme again. Then DNA polymerase will come here and polymerize in this direction. That is from 5' to the 3' direction, right? So this is the case. 3' का template है, 3 to 5' में उसका synthesis कहाँ हुआ? 5' to 3'. And now again the DNA strands will unwind more, again the same process will occur. Again more DNA helices will come and unwind the DNA double helix. more DNA strand will form like this. So the fragments which are formed in the opposite strand, or we can say the strand which have the different polarity than the first one, will have the synthesis of discontinuous strands. So these you can see as these are breakdown, because every time DNA polymerase have to come by the another primer and then synthesize. Again it will come and synthesize. So this is how the stretch, small stretch of DNA strands are forming. So these stretch, the small stretch of DNA strands which are forming on the another template is known as Okazaki fragments. Okazaki fragments. Remember this, Okazaki fragments are present on this template DNA parental strand which is having the polarity from 3' to 5' in the another strand like this. So if the diagrammatic representation has been come, so please remember that what is the activity of DNA polymerase and how the Okazaki, on which template strand the Okazaki fragments are forming. Later on these fragments will get joined by another enzyme. The last enzyme which I have told you is DNA ligase. So this enzyme will help in the formation of simple continuous strand by ligating them together. Now one very important question I want to ask. We have studied the master enzyme which is DNA polymerase and this enzyme DNA ligase. Tell me what kind of bonds they are forming. Okay? Write down in the comment section. So this is how the complete process of DNA replication takes place. Right. Now let's get back to the question. Here certain statements are given on DNA replication. Let's see. The first one is DNA polymerases on their own cannot initiate DNA replication. So yes, it is absolutely true that DNA polymerase cannot initiate the process by its own. उनको एक stepping stone चाहिए था which is provided by primer. Okay, so this statement is absolutely correct. Next one is replication initiates randomly at any place in DNA. So this is Arbina. No, you are incorrect. Those are not hydrogen bonds. Those are phosphodiester bonds. Yes, Bikram, Kormakar, yes, absolutely true. Both DNA ligase and DNA polymerase are forming phosphodiester bonds. Okay. Now, very good. Now, next one is B. Replication initiates randomly at any place in DNA. So, no, this is not the case. Initially, we have studied that kaise origin of replication will help for initiating the process. So, this is incorrect. Next one is replication of DNA occurs at specific phase of cell cycle in eukaryotes. So, yes, this is also true. That specific phase is S phase. So, this statement is also true. Next one is polyploidy results if cell fails to divide after DNA replication. अब जैसे DNA replication हो गया, that means DNA content has been doubled. So आपने वो cell cycle देखा होगा G1S, G2 and then M. So after DNA replication, cell का divide होना ज़रूरी है unless it will lead to the condition which is known as polyploidy. That means the amount of DNA got double but cell doesn't get divide, right? So this statement is also true. ये वाली condition arise होगी if cell fails to divide after DNA replication. Right, so among 4 statements, 3 statements are true. Those are A, C and D, and it is correctly depicted in option 3. So this is how we can hit the correct answer. Okay, now let's proceed to the next question, that is question number 1 to 5. Again, from the molecular basis of inheritance, as I have told you that this chapter is really important from exam point of view. Please read and understand each and every process in this chapter. Okay. Now let's see that this question is basically based on the transcription. Let's read it. Regarding the transcription in bacteria, which statement given below is incorrect? So here the complete process of replication. Let's read about it. Okay. So transcription. let's see what central dogma is. Let's start our process from central dogma. कि DNA था, DNA has the capability to self-replicate, वो just हमने देख लिया that it done by DNA replication, the complete mechanism and the ingredients which are required in that process, and then DNA get form into another molecule which is known as RNA by the process known as transcription. So the formation of RNA on the basis of DNA is known as transcription. These two processes, DNA replication and transcription, both are governed by rule of complementarity. Please remember that. Next, RNA will form the protein by the process known as translation, where protein will be formed on the basis of type of RNA formed, which will be taken by in some another question, obviously. So now in this question, we are going to study about this process. Okay, now what do you understand by gene? Let's start from gene. Gene is unit of inheritance, okay, and it has the capability to code for the protein. For protein, there must be formation of RNA, right? So gene is basically the stretch, or we can say the sequence of DNA which has the capability to form the mRNA, or we can say RNA, and which will further translate and form the protein. So what we can say gene is, it has the functional capability, right? But the complete DNA genome cannot act as a gene, so only very small amount of complete genome is actually acting as a gene. So the lot amount is actually junk, okay? Now let's see, gene could be of two types. One is structural, another is regulatory. For regulatory part, we will study lag operon in some another question. Let's see the structural. So if we talk about the structural, these could be of two types again. Let me expand this. Either they will be present in the arrangement of a split or non-split, right? Split, that means they are interrupted by certain sequence. Split से मतलब समझ में आता है, कट लगे हुए, they are interrupted by certain sequence. So if we talk about eukaryotes, so here if we talk about this gene, so they are basically interrupted by certain sequence which are known as introns. Introns. ये वाला भी हम ले लेते हैं intron. Okay, let me change the pen. Introns. So introns are the basically region which are not going to express any way, right? Please remember that from this thing from I say intron, I say inactivated, like that way. Now this region is exon. This region is also exon. These are coding sequence. Exon start from E. Remember that exon E express. यानी कि exon वाला region ही express करेगा. So this is split arrangement, and this type of arrangement is found in eukaryotes. हम में split gene arrangement होती है. If we talk about non-split gene arrangement, it is found in prokaryotes. Okay, so this is again the differentiating feature between the eukaryotes and prokaryotes once we have read in the ORI, right? Now let's proceed into the process of transcription. If we talk about the bacteria, all the type of RNA get transcripted by a single enzyme that is known as RNA polymerase. If we see the complete name of RNA polymerase, it is DNA dependent RNA polymerase. That means this enzyme is dependent on the DNA, which will be acting as a template, and it will form the RNA, right? And if we talk about the feature that how is going to proceed the process, it's again 5' to 3'. So this is the similarity with the DNA polymerase. Please relate these topics like this way only. ओके सो आरएनए पॉलीमरेज की प्रायोरिटी क्या है, एक्शन किस डायरेक्शन में होता है, इट इज फ्रॉम 5 प्राइम टू द 3 प्राइम. इफ वी सी द स्ट्रक्चर ऑफ आरएनए पॉलीमरेज, it is made up of 5 different polypeptides, 5 polypeptide, which is alpha alpha, these are same, then beta, and then beta dash, along with that omega. This enzyme, when all the 5 polypeptide is present, is known as core enzyme. Okay, so this is the master enzyme of transcription in bacteria. This enzyme is required. Along with that, template is required. Here template is DNA. Along with that, substrate is required, which is ribonucleoside triphosphate. So substrate, जैसा कि हमने replication में देखा था, they are deoxyribonucleoside triphosphate. So here ribonucleoside triphosphate. न्यूक्लियोसाइड ये अलग है, ऐसे समझ लीजिएगा, एंड दिस इज ट्राइफॉस्फेट. राइट, सो सबस्ट्रेट इज दिस. अगर हम इस एंजाइम की एक और बात करें, here the unwinding is required, which is facilitated by this enzyme only. So RNA polymerase can do unwinding of DNA double helix for the process of transcription. So यहाँ पे इसकी दो एक्टिविटी आ गई, एक polymerization की, एक हो गई unwinding की. And the main activity of RNA polymerase when it is a core enzyme is elongation. It can proceed the process of elongation. Okay, along with these ingredients or requirements, another molecules, or we can say factors, are required in the process of transcription. Those factors are sigma factor, which will help in initiating the process, and another one is rho factor, which will help in terminating the process. So these factors are involved. So these are ingredient. The master enzyme is RNA polymerase, which has the 3 main features. Unwinding, it can do unwinding the activities from 5 prime to the 3 prime. Next is it can facilitate the process of elongation, right? Now let's see the actual process of transcription. So this is basically the transcription unit, and this is the complete process of transcription. Let's see that how it is going to proceed. The master enzyme here in this process is RNA polymerase, right? When only 5 different polypeptide is present, then it is known as core enzyme. Then we have talked about certain factors which are sigma and rho. So when RNA polymerase get bind to the sigma factor, it will able to initiate the process. So this factor will help in initiating the transcription process, and then the enzyme will be known as holoenzyme. Okay, please don't get confused. What is holoenzyme? Holoenzyme, RNA polymerase along with a factor, then it is known as holoenzyme. RNA polymerase itself having the 5 different polypeptides, then it is known as core enzyme. Right. RNA polymerase की जो itself activity है वो है elongation करने की. So it can elongate the process, or we can say elongation of RNA from the DNA molecule can itself be done by RNA polymerase. For that it do not require any factor. So once the sigma factor will attach to the RNA polymerase, it will initiate the process and then it will get removed. And the core enzyme, which is RNA polymerase, is able to facilitate the process of polymerization of RNA, right? So it is basically able to do the process of elongation itself, जब ये सिर्फ core enzyme है. Okay. Now next one is when another factor, which is rho factor, will come into play. it will again form the holoenzyme. किसी भी factor के साथ ये जुड़ जाएगा, then it will form the holoenzyme and it will remove, or we can say that terminate the process of transcription by removing the RNA polymerase, and after that nascent RNA will be fall off like this. So this is how the complete process of transcription takes place in the bacteria. Please remember that RNA polymerase along with sigma factor is able to initiate the process, but RNA polymerase alone can do the process of elongation, right? And when it is again attached with the rho factor, then it can terminate the process. So अगर हम activity के बारे में बात करें, तो अगर हम एक्टिविटी के बारे में बात करें तो आरएनए पॉलीमरेज कैन इटसेल्फ इनिशिएट द प्रोसेस ऑफ इलॉन्गेशन ओनली, बट उसको फैक्टर्स चाहिए टू इनिशिएट एंड टर्मिनेट द प्रोसेस. ओके, सो दिस इज द कंप्लीट प्रोसेस ऑफ ट्रांसक्रिप्शन इन बैक्टीरिया. अब हम इसकी बात कर लेते हैं व्हाट इट इज. पेन, लेट्स सी. So this is basically the transcription unit in which the transcription will be started or done. So this is the template strand. This yellow one is the template strand. And this green one is coding strand. If we see the polarity, 3 prime, 5 prime, as you know that it moves from 5 prime to the 3 prime direction. Arrow is already given from this to this, right? RNA polymerase को attach होने के लिए required certain position which is known as promoter. So promoter is the region where RNA polymerase will get attached to the transcription unit and initiate the process. So this is basically the attachment site for the RNA polymerase, and the DNA and the transcription will be started from here, not from the promoter region. This is the structural gene. Structural gene. Structural strand, right? Which we have studied in the initial of this question, जहाँ पे बताया था कि structural gene और regulatory gene होते हैं. तो this is the structural gene which will code for the RNA. So इसको template strand कहते हैं, इसको coding strand कहते हैं. Template strand will act as a parent on the basis of which the RNA will be formed. That means the RNA formed will be complementary to the template. As I have told you that it follows the rule of complementarity. So it will be complementary to the template strand, but it will be similar to the coding strand except on the position of thymine it will have uracil. That's why it is known as coding strand. It does not have any role in the during the transcription process, but it is known as coding strand because the stretch of RNA which will be formed from this template strand will be having the same sequence as of coding strand, but on the position of thymine they will have uracil. Okay? So this is how the transcription unit works or looks like. On this also certain question has been asked. The representation of transcription unit will be given and you will be asked that ये structure क्या है और ये structure क्या है. This is terminator. Terminator. Remember that promoter is present upstream to 5 prime strand. Now let's get back to the question which says which statement is incorrect with respect to the transcription in bacteria. First statement, there are 3 types of DNA dependent RNA polymerase for this process. So this is incorrect. The first statement is incorrect. RNA polymerase is the only enzyme in the prokaryotes which can facilitate the polymerization of RNA, any type of RNA polymerization के लिए mRNA, rRNA और tRNA, only one enzyme is required, that is RNA polymerase in bacteria. So first statement ही incorrect हो गया. If we talk about eukaryotes, then there 3 types of RNA polymerase is required. Basic 3, more are also present, RNA polymerase 1, 2 and 3, but those are in eukaryotes. If we talk about prokaryotes, that is bacteria, वहाँ पे सिर्फ एक RNA polymerase की requirement है. So पहला ही statement incorrect हो गया. Let's see the second one, which says mRNA does not require any processing to become active. Yes, bacteria की बात हो रही है. As I've told you, the site of transcription in bacteria is cytosol. So वहाँ पे कोई requirement नहीं है to further modification की, so that it can move from the nucleus to the cytoplasm like that way, जैसा कि हम eukaryotes में देखते हैं. For example, the splicing, polyadenylation, poly-A tail, etc. All those things will be occur in eukaryote but not in prokaryotes. So ये statement correct हुआ. Statement 3, it takes place in the same compartment where translation occur. Obviously, no compartmentalization is present in the prokaryotic organism. So, दोनों चीज़ एक ही जगह पर होगी, that is cytoplasm. Next one is, many times the translation can begin much before the mRNA is fully transcribed. So, yes, it is obviously true because कोई modification तो चाहिए नहीं in bacteria में, तो इसलिए as the question is talking about the incorrect statement, so here we can rule out option 2, 3 and 4 and hit the correct answer, that is option 1. I hope I made you understand these topics very clearly and I'm able to, sow the seeds of knowledge in your brain and hope it will bloom wherever you want. Thank you for such a patience. Please stay tuned for the next session. Thank you so much.